Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore two fascinating and critical concepts, aneuploidy and polyploidy. Imagine your DNA as a highly detailed instruction manual. What happens when a single page is missing or an entire chapter is duplicated? We will discover how these numerical changes in chromosomes occur, how they affect living organisms, and why they are responsible for both severe human genetic conditions and the robust fruits and vegetables we eat every day. Let us dive in.
To start our journey, let us imagine you are building a massive Lego castle using a specific instruction manual that comes with exactly 46 bags of pieces. Now, imagine that by mistake, the factory forgot to put bag number 21 in your box, or perhaps they accidentally gave you 2 copies of it. The vast majority of your Lego set is perfectly fine, but that one specific missing or extra bag is going to cause noticeable problems when you try to build the castle. In genetics, this exact scenario happens when a cell ends up with an extra chromosome or is missing one specific chromosome. We call this specific type of genetic typo aneuploidy. It is not a mistake involving the whole instruction manual, just one specific chapter or bag of parts.
Now that we understand what aneuploidy is conceptually, let us look at how it actually happens in a cell. Normally, most animals and plants are diploid, meaning they have 2 copies of every chromosome, one inherited from each parent. However, when an error occurs during cell division, specifically a process called non-disjunction, chromosomes fail to separate properly. As a result, a sperm or egg cell can end up with the wrong number of chromosomes. If an organism is missing one chromosome from a pair, it is called monosomy. If an organism has an extra chromosome, making 3 copies instead of the usual 2, it is called trisomy. Because chromosomes contain hundreds or thousands of genes, having an extra or missing chromosome creates a massive imbalance in the proteins the cell produces, which is why this condition is so impactful.
This biological imbalance we just discussed can be represented mathematically by modifying the standard diploid chromosome number. The formula for aneuploidy is C aneuploid equals 2n plus or minus x. Let us break down where this comes from. The term C aneuploid represents the total number of chromosomes in the affected organism. The term 2n represents the normal diploid number of chromosomes for a specific species, where n is the basic set of chromosomes. Finally, the variable x represents the number of individual chromosomes that have been gained or lost. If x is 1 and we are adding it, the formula represents trisomy, meaning 1 extra chromosome. If we subtract it, it represents monosomy, meaning 1 missing chromosome. When the value of x increases, the genetic imbalance becomes more severe, often becoming fatal.
Before we move on, let us see this formula in action with a concrete example. The problem states that humans normally have a diploid chromosome number of 46. We are asked to find the total number of chromosomes in a child born with Patau syndrome, which is caused by trisomy of chromosome 13. First, we identify the normal diploid number, which is 46. Next, we recall that trisomy means there is 1 extra copy of a single chromosome. Therefore, we choose the formula for trisomy, which adds 1 to the normal diploid number. We substitute 46 into the equation and add 1. This gives us a final answer of 47. This single extra chromosome disrupts the delicate balance of gene expression, leading to the severe developmental challenges associated with Patau syndrome. Let us look at another example, this time involving the loss of a chromosome. We are given that the fruit fly,
Drosophila melanogaster, has a normal diploid chromosome number of 8. A geneticist discovers a mutant fly with monosomy for chromosome 4, and we need to find its total chromosome count. We start by noting the normal diploid number is 8. The problem states the fly has a monosomy, which means the loss of one single chromosome from the normal diploid set. Because it is a loss, we use the monosomy formula, subtracting 1 from the total diploid number. We substitute 8 into our equation, subtract 1, and get a final value of 7. Because this fly is missing a chromosome, it will likely show altered physical traits or reduced viability compared to a healthy fly.
To truly grasp these numerical changes, let us look at a visual representation of these chromosomes. On the screen, you are seeing a comparison of 3 different cell types. On the far left is a normal diploid cell with a total of 6 chromosomes arranged neatly in 3 pairs. In the middle, we see a cell exhibiting monosomy. Notice how the 3rd pair is incomplete. It is missing a chromosome, bringing the total count down to 5. On the far right, we have a cell exhibiting trisomy. Here, the 2nd group has an extra 3rd chromosome, bringing the total count up to 7. This diagram clearly illustrates how aneuploidy affects just one specific set of chromosomes, leaving the rest of the genetic material in its normal paired state. Now that we have explored the gain or loss of individual chromosomes, let us shift our focus to a much larger scale alteration. Let us go back to our Lego castle analogy. Instead of missing just one bag of pieces, imagine you open your delivery box and find that the factory accidentally sent you 2, 3, or even 4 complete sets of the entire 46-bag Lego kit. You do not have an imbalance of parts, you just have way more of everything in perfect proportion. In the biological world, this is exactly what polyploidy is. Instead of gaining or losing a single chromosome, an organism ends up with entirely extra sets of all its chromosomes. It is a massive scaling up of the genetic instruction manual, resulting in organisms that are triploid, tetraploid, or even hexaploid. This massive scaling up of genetic material has very different consequences depending on the organism. Polyploidy can be categorized into autopolyploidy, where all chromosome sets come from the same species, and allopolyploidy, where the sets come from different but closely related species hybridizing. While polyploidy is generally lethal in humans and most animals, it is incredibly common and highly beneficial in the plant kingdom. In fact, many of the fruits and vegetables we eat every day are polyploids. Having multiple sets of chromosomes often makes plants larger, more robust, and capable of producing bigger flowers and fruits. Furthermore, farmers use polyploids with an odd number of chromosome sets, like triploids, to their advantage. Because their chromosomes cannot pair up evenly during reproduction, these plants are usually sterile, which allows us to create seedless varieties of fruits like watermelons and bananas. Because polyploidy involves entire sets of chromosomes, the math is quite straightforward. The formula is C polyploid equals X times N. Let us define these terms. C polyploid represents the total number of chromosomes in the polyploid organism. The variable N represents the monoploid number, which is simply the number of chromosomes in a single basic set. Finally, the variable X is an integer greater than 2 that represents the ploidy level, such as 3 for triploid, 4 for tetraploid, and so on. Conceptually, this formula shows that we are just multiplying the entire basic genetic code. When the value of X increases, the total amount of DNA in the cell increases proportionally. In plants, this massive increase in genetic material often leads to larger cell sizes and, consequently, larger physical traits.
Let us apply this formula to something you might find in your grocery store, a seedless watermelon. We are told that a normal, seed-bearing watermelon is diploid, with a total of 22 chromosomes. Agricultural scientists create a seedless watermelon by breeding it to be triploid. We need to find the total chromosome count for the seedless variety. First, we must find the basic set number, n. By dividing the normal diploid number of 22 by 2, we determine that a single basic set contains exactly 11 chromosomes. Next, we identify the ploidy level. Since it is triploid, x equals 3. We multiply the ploidy level of 3 by the basic set number of 11. This gives us a final answer of 33 chromosomes. Because 33 is an odd number, these chromosomes can't divide evenly, making the plant sterile and seedless.
Let us look at one more example, this time involving a massive accumulation of genetic material. Modern bread wheat is an allohexaploid, meaning it contains 6 complete sets of chromosomes derived from 3 different ancestral grass species. We are given that the basic chromosome set for these ancestral grasses is n equals 7. We need to find the total chromosome number in modern bread wheat. The problem clearly states the basic monoploid number is 7. It also tells us that bread wheat is hexaploid. The prefix hexa means 6, so our ploidy level, x, is 6. We simply substitute 6 and 7 into our polyploidy equation. Multiplying 6 by 7 yields a final answer of 42 chromosomes. This massive accumulation of genetic material gives bread wheat the robust traits and high yield that make it a vital global crop.
To visualize how polyploidy scales up an organism's genetic material, let us look at this diagram comparing different plant cells. On the left, we have a standard diploid cell, labeled 2N, containing 2 complete sets of chromosomes. Moving to the middle, we see a triploid cell, labeled 3N. Notice that it contains 3 full sets of chromosomes, and the cell itself is drawn slightly larger to accommodate the extra DNA. Finally, on the right, we have a tetraploid cell, labeled 4N, containing 4 complete sets of chromosomes. This cell is the largest of the 3. This visual perfectly captures how polyploidy maintains perfect genetic proportion. There are no incomplete pairs like we saw in aneuploidy, while significantly increasing the total volume of genetic material and often the physical size of the cell itself. We have covered a lot of ground today, so let us review the key takeaways. First, we learned that aneuploidy is the gain or loss of individual chromosomes. This creates severe genetic imbalances, resulting in conditions like monosomy and trisomy, which are often harmful or fatal in animals. Second, we explored polyploidy, which is the multiplication of entire, complete chromosome sets. Unlike aneuploidy, polyploidy maintains perfect genetic balance. Finally, we saw how polyploidy is a major driving force in plant evolution and agriculture, allowing us to cultivate larger, more resilient crops and seedless fruits. You now have a solid understanding of how these numerical changes fundamentally alter the genetic instruction manual of life. Great job following along.